This is a story about lipoprotein lipase. Lipoprotein lipase, otherwise known as LPL, is secreted by cardiac, adipose, and muscle tissue. It hydrolyzes triacylglycerides in lipoproteins. The amino acids that make up LPL's active sites are aspartate, histidine, and serine. LPL acts on a substrate called triacylglyceride. In order to activate this enzyme, ApoC2 is needed as a cofactor. Triacylglycerides, along with cholesterol, ApoE, and ApoC2, make up what is known as a chylomicron. ApoC2's active site is what is known as lysine 147 and lysine 148. This is just assumed, however, not proven. An oxyanion hole is also present within LPL. This is made up of tryptophan and leucine. The product formed in the hydrolysis of triacylglycerides by LPL is what is known as free fatty acids. These free fatty acids end up in tissue cells and are then used for energy. Welcome to the cortical empire. Cortical is short for cardiac, adipose, and muscle tissue, as within this tissue is where our story takes place. We start with our active site, aspartate, histidine, and serine, boarding the boat the lipoprotein lipase, as this marks the beginning of their fishing adventure. The lipoprotein lipase sets sail and is secreted from the cortical empire. The ship moves through the water, heading towards capillary channel. "We're here!" yells the excited amino acids. The lipoprotein lipase continues down into capillary channel to find somewhere to stop and fish. "Cast the rod!" yell the amino acids. A fishing rod is cast into the channel to test the waters. The bait used at the end of the rod is lysine 147 and lysine 148, known for catching cofactor fish, specifically ApoC2. Underneath the surface of the water is a school of fish, otherwise known as a chylomicron, containing cholesterol, ApoE, ApoC2, and triacylglycerides. Cholesterol and TAGs have swum from the dark depths of the small intestines, whereas ApoE and ApoC2 came from other streams, specifically bloodstreams, to form this chylomicron. The lysine 147 and lysine 148 bait immediately catches the eye of the ApoC2 fish. The lysine 147 and lysine 148 line is reeled back up to the lipoprotein lipase deck. "Hooray!" cheered the crew after reeling up an ApoC2 fish. Larger fishing nets are then cast into the water in the hopes that more fish from the chylomicron are caught. The fishing nets are then pulled up and unloaded on the deck of the boat right at the amino acids' feet. All the fish that were caught are TAGs, exactly what the amino acids were looking for. Serine picks up a TAG fish and gets to work, starting the enzyme-substrate reaction and degrading the TAG. To start off the reaction, aspartate forms a hydrogen bond with histidine, allowing histidine to become basic and deprotonate serine. The serine then acts as a nucleophile and attacks one of TAG's carbonyl groups on the glycerol backbone. This creates a negative tetrahedral intermediate. Suddenly, the negative tetrahedral intermediate is scooped up by an oxyanion bird. Inside the oxyanion bird, the tryptophan and leucine help to stabilize the negative tetrahedral intermediate. After the intermediate is stabilized, the oxyanion bird drops the intermediate and flies away, continuing the reaction. The tetrahedral intermediate can reform the carbonyl group and recreate the glycerol backbone, which then acts as the leaving group, forming a diacylglycerol. A water molecule from off the TAG fish is then used to donate a proton to the histidine, which creates a reactive hydroxyl anion. This hydroxyl anion attacks the carbonyl carbon, forming another negative tetrahedral intermediate. "Oh no!" screamed the active site amino acids. The oxyanion bird was back again to stabilize the negative tetrahedral intermediate. Inside the oxyanion bird, the tryptophan and leucine help to once again stabilize the negative tetrahedral intermediate. After the intermediate is stabilized, the oxyanion bird drops the intermediate off again and flies away. The tetrahedral intermediate then reforms to a carbonyl, and the catalytic serine is released. A free fatty acid is formed as a product of this reaction. After the free fatty acids are formed from the TAGs, the lipoprotein lipase can set sail once again. The remnants of the chylomicron are released back into the capillary channel. The diacylglycerols and cholesterol swiftly make their way to liver falls, where they are further metabolized. ApoC2 and ApoE make their way back to high-density lipoprotein land so that they can help make new chylomicrons. After returning to Cortical Empire, the free fatty acids are brought to town to be traded and sold as a source of energy to cells. The end.